Great pleasure to introduce David Foster to give the first research talk of the afternoon. David has kind of a perfect education also. He started off in studying physics at Imperial College in London. He then moved on to work with kind of the two greats in the field. One, the empiricist Richard Morris, who, among other things, invented the water maze, but is known for many decades of brilliant empirical studies of the hippocampus, and was co-advised by theoretician Peter Dan in his PhD at the University of Edinburgh. He then continued his training as a postdoc at MIT in Matt Wilson's lab, yet another game-changing lab. I think all of this contributing to David's incredibly creative, interesting work, very different from anyone else, but you can kind of see all the different strains of these mentors in influencing his the kinds of ideas he asks. But I think it really is a kind of perfect training with a background in physics and then working with Morris, Dan, and Wilson, because he's really addressing the really hard questions, the really cognitive questions of the hippocampus. What is it really doing? What is it really thinking? How much more complex and profound is the story of play cells going to get? And I think David is he's the person who's going to be the one to find this out. And I won't steal any thunder because he's about to tell you, but I think if by the end of his talk, you're going to feel the way I do, which is that we're really standing on a tip of a scientific iceberg when it comes to the hippocampal play cell. Thank you.
Well, thanks, Lucy, for inviting me, and to the department for inviting me and for that introduction. And it's true, I can't blame any of my former advisors if I fail to match up. Right. Well, let's start with this. You're a rat and you're in a maze. How do you get out? So you're going to find your way from, this is a real maze, right? It's a kind of difficult maze. You find in a puzzle book. First question, can everybody hear me? So, just to remind you, you're a rat in a maze. You've got to navigate through the maze. What's difficult about this maze? Well, the first thing I would say is the food's very far away. So you may have no direct information. You can't see or smell the food that can get you there. Also, in terms of trying to explore here and associate responses with the food, what's going on over here is very far away from what you need to do at the beginning. The second problem is that even if you actually knew the direction, the vector from your start to your finish, if you could actually smell the food and guess where it was, that wouldn't actually help you, because you need to take a different set of decisions in order to find your way through the maze. So it's a bit like if I want to get to the bell tower and I know it's there, that gives me only limited information. It could be very useful, but I also need to know that I need to go take that action and then a different action and a different action in order to get there. So there's an element of navigation that involves understanding not just space, but on top of space, the contingencies that are presented to you by the environment. So I won't repeat this because you actually heard it already, but Tolman hypothesized that there should be a field map of the environment, something like a field map of the environment in the brain. And it's this idea of a cognitive map that we're all going to be talking about. So let me introduce the real star of this talk, at least, which is the hippocampus, here shown in the rat. And it's a very similar structure in the human brain, but at a different orientation. It's one of the best studied areas of the brain, but what I'm going to be telling you about in particular is, in this talk, and there's a lot of interesting things that I could tell you about this circuitry, but I'm just going to tell you about the activity of these units in the output area CA1 of the hippocampus. And I'm going to tell you information that we've gathered by dropping electrodes into that area of the brain and recording the activity of these units in animals that are actually able to freely move around. So of course that's sort of what we've been talking about, but I want to make sure that everybody understands. So we drop the electrodes in. Actually, it takes several days. We lower the electrodes, and they're nestled in the extracellular space, and they're able to pick up signals from surrounding neurons. We use a trick called a tetrode trick, which is we actually use 4 wires twisted together. Each tip is recording from the same local population of cells, but with a slightly different spatial location. So you can triangulate the signal and separate out physically different sources corresponding to physically different cells that are surrounding this electrode. So from one tetrode, we can distinguish multiple different sources of signal. So we can record the action potentials of a number of cells from a given tetrode, and then we actually implant many tetrodes simultaneously. So the upshot is we're able to record large numbers of cells. Not all the data I'm going to tell you about actually use that tetrode technique. There's been a developing technology, but I'll just want to recap. So the fundamental feature here is the place cell phenomenon. So what happens in the hippocampus of a rat when a rat's running up and down a track like this? Let's say this is about 2 meters, a 2-meter long track. What I'm showing you is the positions where 3 different CA1 cells fire spikes. So I've just color-coded them as a green cell, a blue cell, and a red cell. And this is simultaneously recorded in the same animal, running up and down. He might have run up and down something like 20 times. And what you can see is that the green cell fires clearly, preferentially in this area, the blue cell in this area, and the red cell in this area. And so this is your classic place cell activity. And these cells can also be directional, so they will often prefer to fire when the rat's moving in one direction over the other. It was the discovery of cells like this by O'Keefe and Ostrovsky in 1971, which prompted Lynn Nadell and O'Keefe to make the claim in 1978, as he just said, that these cells instantiated a cognitive map in the brain. And I want to explore that idea. I mean, it's clear that this idea, if people don't know already, this is the Nobel Prize winning idea together with Edvard and May-Britt Moses' grid cell discovery. And so, yeah, they got it right. But I want to explore some of the consequences of this and how it relates to navigation. So this could be a map, but just taken at face value, there's a problem. Let's say you're a rat in the maze. What are the kind of things you might want to do? Well, you might want to, if you get somewhere interesting, think, how did I get there? Or you might be at a particular point in the maze and think, what am I going to do to get out of here? Place cells, as I've described them so far, tell you where you are, which is a funny kind of map where you can only look at the current location. What you really want to do, of course, is escape from your current location and look around. So I'm going to say, map out a few properties that a true Tolmanian cognitive map should have. If you're in this maze and you're trying to get out. The first thing is you should be able to access places away from the current location. Right? That's pretty clear, because if you were looking at this map, that's what your eyes would be doing. They'd be scanning around this map to look at how you can get out. What you're doing when you're looking through the map is simulating your experience. That simulation of experience had better reflect the real spatial contingencies in the environment. It's no good if you can move away from your current location, but you can zip around teleporting from one location to the other. That's not going to be a good simulation for gauging your navigational actions. You need to be able to have your internal simulations respect the spatial constraints of the environment. And the third thing, which comes directly from Tolman, is your simulations should have a goal. Presumably, if you're trying to get out, you should be able to say, how do I get to this particular position? Or how do I find the food? So I'm going to explore each of these in turn and give you some evidence. So Lynn talked about this a little bit, but it's long been known that in the hippocampus there are different modes, activity modes, depending on behavior. So for example, here we have the local EEG, in left and right hippocampus, which is giving us, this is activity that reflects the combined activity of thousands of cells and also synaptic events. But this is the substrate for recording brain waves, oscillatory activity. And here you see this characteristic, so this is a second, this is about half a minute. Here we see this characteristic 8 Hertz theta activity that occurs when animals are running around. But everything we know about place cells, the fields, is recorded here, right? But as soon as the animal stops moving, the theta disappears, and the hippocampus enters this sharp wave mode, where there's relative quiet, punctuated by periodic depolarizations, actually, of lots of cells altogether, which show up as these sharp waves in the EEG. And if we zoom in, we can actually find high-frequency oscillations known as ripples that ride on that envelope. So this whole thing's called a sharp wave ripple. And so essentially, for the whole talk now, I'm going to be telling you about what place cells are doing in these sharp wave ripples, which really complements and adds to what we know they're doing in this state. And I should mention one other thing. So place cells were first characterized by recording one cell at a time and correlating that activity with behavior. What I'm going to be doing now is telling you what happens if you record large numbers of cells simultaneously, because if the cells are doing interesting things, like scanning the environment and moving away, I'm not going to be able to see it by correlating that activity with anything in the world. I'm going to have to see it in the coactivity of cells talking to each other. That'll become clear. So let me take you through a very simple experiment. A rat running up and down a 2-meter track. So in gray, you can see the behavior of an animal over a course of 20 minutes, running up and down about 2 meters of track. So he sweeps back and forward from one end of the track to the other, as you can see in gray. He also pauses for a few seconds at each end. Superimposed, you can see the times and positions of spikes from a single CA1 place cell. And as you can see in this example, this cell fires when the animal rounds the turn in this direction. So this is your classic place field. And you can capture that with histogram normalized by the occupancy. Across a set of cells recorded simultaneously, you'll find something like this. 19 cells recorded simultaneously. They each show a preferred firing location, but collectively they span the track. If you like, this is our map across these cells for this track. So now I want to use this template, this population, to actually look in very great detail at just one single lap of experience. And that's what you're going to see here. So now we've got an animal moving from one end of the track to the other, and it takes a few seconds. This is time. And he moves from one end of the track to the other, and then he stops, absolutely still, like just grooming and doing some other things, but not moving, not running anywhere, at one end of the track for the rest of 1 minute. I'm going to show you what these 19 cells did. It's all recorded simultaneously. As the rat runs through the cell's place fields, you get this classical place field activity, where the cells are firing in an order which is given by where their place fields are. But when the animal pauses, you find that these cells are active because that's the animal is currently in those cells' place fields. But you also get these depolarizations, these population events, where large numbers of cells seem to be firing simultaneously. If we zoom in on just one of those events, I just expand the time x-axis, what you find is these events are actually structured, in this case, firing in a sequence which is the reverse of the sequence that was experienced when the animal was running. So the cells that have firing fields close to where the rat is fire first, and then backwards, recruiting all the other cells until you get to the beginning of the track. The animal's stationary throughout this. This whole thing takes on the order of 100 milliseconds. And this is happening this is the EEG filtered for the ripple band. This is happening during the ripple. So this is what sharp wave ripples are. They're I have to show you more evidence, but they're sequential events. Just to give you an idea of how sort of repeatable this is, these are the events that were seen just for each lap on this track. And so this is if we go back, you see there's something like 12 events here, and they each had this profile. So in each case here, it's the same 19 cells on the y-axis and the same, say, 220, I think, milliseconds on the x-axis. And you see it's very regular firing each time. So it seems like a very efficient way to use experience. You run once, and then you have multiple replays. So let me back up and say that the first demonstrations of this kind of activity were in sleep, but we found this activity was very robustly observable during awake behavior whenever an animal pauses. And that awake replay has now been replicated in multiple labs, and I'll just draw attention to 3 results in particular. Firstly, it's now known that during the entire episode of pausing, running, and pausing again, you can get forwards-going replays at the beginning of the trajectory, and then reverse-going replays at the end. That's a typical pattern. Fascinatingly, if you make the track very, very long, in this case a 10-meter-long track folded around, the replays will, as I think I mentioned before, they don't tend to jump, but they will happily chug along at a fixed mental speed, so that the replays in this case can last 600 or 700 milliseconds. And finally, a fascinating result from Lauren Frank's lab showed that if you try and interfere with sharp waves by using electrical activity to disrupt activity in the hippocampus contingent on detecting a sharp wave, you can have a very selective impairment in behavior. So in an alternation task where the animal has to alternate left and right on this track, the incoming behavior is unaffected. So if you disrupt sharp waves at the beginning, I said there were forwards events, but if you disrupt them at the beginning, the incoming trajectory is unaffected, because that's something the animal does every single time. He's learned somehow that he doesn't need to go down there. But the choice point where he has to alternate, if you disrupt activity at the beginning here, then he can't perform the alternation anymore. So somehow the flexible generation of behaviors is particularly dependent on these sharp wave ripple events. All right, so thinking of Tolman, there's one point in particular I want to draw your attention to, which was one of the criticisms of Tolman's work at the time by one of his contemporaries, Guthrie, who said, in his concern with what goes on in the rat's mind, Tolman has neglected to predict what the rat will do. So far as the theory is concerned, the rat is left buried in thought. If he gets to the food box at the end, that's the rat's concern, not the concern of the theory. This, and everybody remembers this, I think, as lost in thought. So this was really a criticism of the theory that Tolman wasn't accounting for the exact mechanism for producing behavior. But it's actually something that's always, I've always taken it as a potential criticism of this replay as well. So here we have something where we're claiming that there's an offline production of sequences, but you might be skeptical that the rat would ever have time to employ this procedure if he's actually running for his life or he's desperate to find his way out of a situation. So I think that's a valid criticism, and I'm going to show you 2 ways that can be addressed. The first is to point out that the replay sequences are temporarily compressed. So an experience that might take many, many seconds, 10 or 20 seconds, can be replayed within just a fraction of a second. So that's a feature by which extended experience can be processed quickly. But a second way to think of this is that indeed it may be that in some cases replay exists to set up an understanding of space which can then be accessed very, very quickly during the production of behavior. And I'm going to give you one example. So if you actually have this difficult maze, and you're sitting here, and this is actually your traffic pattern, and you get your GPS to calculate your path, the chances are the GPS is going to use an efficient algorithm to do this. The GPS will know what you might call the transition functions. It will know this map of what leads to what. And an efficient algorithm called a flooding algorithm will start at the end where the goal is and run back the numbers of steps to develop this thing called a value function. Why does it go backwards? It's taking advantage of the Markov property of this navigation problem. The Markov property is that what you actually have to do at any given location only depends upon that location, not how you got there. In other words, at this point, it doesn't matter if I came from this direction, or this direction. The right thing to do is to go in this direction, right? That means that if I start at the last location, then I know that the preceding one is going to want to go in a particular direction, and I can ignore everything else. So that gives me an efficient way to assign, in this case, numbers of steps, costs, value, it's interchangeable, to every location efficiently. And this algorithm only has to visit every location once. If I try and plan in the forwards direction, I have a combinatorial explosion of possible paths that makes it much less efficient. So your GPS will be using an algorithm like this. The point about this is if you have these values, it completely localizes and solves the navigation problem. Now, when you're in the maze, all you have to do at any difficult point is take the choice that minimizes the number of steps. And that's the right thing to do. So you don't have to think anymore. So you're not lost in thought. So we hypothesized that the way this could work is that if reverse replays were paired with a reward signal that has a particular characteristic, has a fast onset and a slow decay, then the result would be that places near to the goal would be associated with high reward, and successively further away places would be associated with successively smaller amounts of reward. And in this way, you could develop a value function over space that estimates, essentially, for every location, that cost function I told you about before. Now, there's a number of different ways to explore this model. What I'm not going to tell you about is evidence that the hippocampus and various value-representing areas might be active together, but that's a very active area of research in my lab and a number of other labs. Instead, I'm going to tell you about this thing. So this says that once you've done reverse replay, you've set up these values, then while you're running along, you should be able to access these values to guide you in your navigation. So I need to back up and tell you one thing, and then I can get back to that. And that is that it turns out that activity is not just sequential when the animal stops. It's sequential all the time. Place cells are active in sequences all the time, but when the animal's running, those sequences tend to be quite short. And this is known as theta sequences, but it was first really discovered by John O'Keefe as a phenomenon called theta precession. Now, it's a little complicated, but let me try and get through it. So the idea is that when an animal runs through one cell's place field, the spikes from that cell are pulsed at theta, but not perfectly at theta. In fact, the actual phase of theta gets earlier and earlier and earlier as you go through the field. And you can actually plot that across multiple traversals as a phase position diagram. And you get this characteristic phase precession, phase reduction as you move in position through the field. So this is position, this is phase. What does that mean? Well, this is a single cell doing exactly that, right? The cell fires at a late phase, earlier phase, earlier phase. But if a whole bunch of cells are doing this, what that means is you actually have sequences. So this phase precession was an early insight into the fact that, in fact, there are these short little sequences that are moving ahead of the animal as he's moving. We verified this directly, however, by recording from larger numbers of cells simultaneously. And here you can see the same 19 cells as before, but now I've actually just zoomed in on the classical place field activity. And so for each cell, there's this place field. If you look horizontally, that's a one cell. There's a place field the animal passes through, but it's also pulsed at theta. And when you look across cells, you can see there are these short look-ahead sequences. Which brings me to this fascinating result from David Reddish's lab, actually Matt Vandermeer's work with David Reddish, which is, to me, it's very exciting, because it says when you record from ventral striatum, which might be thought of as one area where you expect to get value information, as an animal approaches the goal, there are a subset of neurons that show these ramping activities, which is exactly what we would have predicted, increasing the expectancy as the animal approaches the goal. But even better, those cells that show this ramping activity actually show phase precession with hippocampal theta, and not with their own theta. So these cells are firing in a way which indicates that they are taking part in hippocampal sequences. So every time there's a hippocampal sequence, there's these ventral striatum cells are firing at the end of that sequence, as if there's a direct readout at every point of what the value is of taking that next step. But this is an ongoing area. So I want to turn now to the second thing that we want our internal simulations to be, and that is accurately reflective of the spatial environment. So I could examine that very complicated maze, but in fact we can capture many of those issues in a much simpler and reduced maze. So far I've told you mainly about linear tracks, so we're going to move up to a next level of complexity, which is a track with a single choice point. And also one of the arms longer than, twice as long as the others. So what happens when a rat explores a new environment like this? How does the replay encode this environment? So I'm going to show you just a few slides, about 3 slides about this. But I'm going to have to explain to you this linearization. So in order to be able to show you these replay sequences, I take this 2-dimensional thing here, this maze, and map it onto a single dimension. So these positions end up corresponding to this part of the y-axis, then the green positions to the middle, and the blue positions to the top. So here we have about, 100 seconds, so just over a minute. And what the animal did was he started at the choice point, he moved out to the C end of the orange arm, he stayed there for a bit, and then he moved back to the choice point, he moved into the R end of the green arm, stayed there for a bit, and then moved back smoothly into the L arm, okay? We recorded from over 80 cells with place fields distributed throughout this maze. And you can see that, and then I've ordered these cells according to where the peak firing was. And you can see that this activity shadows the behavior quite nicely, except for these intermittent population events, which are going to turn out to be replays. But to look at a cleaner version of this, we can do something called position estimation, where we simply take these spikes and combine them with the knowledge of the fields to say, what's the most likely position that is being represented by the hippocampus? And this really does, so this is just taken from neural data, but it really does match his behavior during behavior. So what do we see for replays? Well, from the very first exposure to this track, after very little experience, just a few minutes, you start to get the first replays, and you get replays of each of the individual arms. But interestingly, you get these replays that cross the arms, as if representing trajectories from one arm into the other. So here's an example of a replay. The animal is still, at this point, at one of the ends. And the replay takes a few hundred milliseconds to cross from the C end into the R end. And you get large numbers of them after just a small amount of experience. Here are replays that cross from the R end, so if we R end into the L end. And we get large numbers of these, or the reverse. And here's the other kind, going from the C to the L, or backwards. So you've got these replays that match putative trajectories in the environment. What you don't see is replays that cross over or do combinations of sequences that don't match any behavior. But we still have one very... this is just to show that these are learned very, very quickly. So it's a little bit complicated, but I'll just show this in this figure. We take for each of the kinds of replays we're talking about, across the stopping periods, we add up in the black the number of replays that we see. And at some point, we've seen enough to say, yeah, we would never get this by chance. So we can already say that after a small amount of experience, 2, 3, 4 laps of experience, you're getting so many replays that we're absolutely confident that this is not anything that would have happened by chance, that we see a replay of that experience. However, this is probably an overestimate of how much experience is necessary, because in this task, it's up to the animal how long he stays at the end. So if we somehow induce the animal to spend a long time, we'd probably see, I would guess we would see replay even after one experience. But in any case, it's very rapid. So this is the point I wanted to get to. We're generating these sequences, right? But there are many different ways that you could generate the sequences that would represent these trajectories. And there's, in particular, there's a sort of episodic way to do it, and there's a Tolmanian way to do it. All right. The episodic way is where you use different cells to represent different episodes of running. So you would say, all right, when I move from the C end to the R end, I use one population to represent that. But then I use a different population to represent each other trajectory. And in that way, you could rapidly assign, that would be a very efficient way to rapidly assign experience to replay sequences. But it wouldn't give you any basis for spatial generalization, because the cells wouldn't know anything about the shape of the maze. Or you could actually build the maze, stitch the cells together, in a sense, to actually reflect the structure of the maze. So we thought about these two hypotheses and said, well, and this is for, I'm just talking about replay. So we know where the place cells are, we know the fields are, sorry. This is just about replay sequences. So we said, right, let's take, for example, the sequences that occur when the animal's at the C end. Sometimes you get sequences going up and to the left. Sometimes you get sequences that go up and to the right. If we look at just the C portion, and if you look under here, there's a red and a blue curve. Just look at the red curve. There's across the 80 cells, there's huge variability in the firing rates. So cells are some cells are firing a lot during these replay sequences, some cells are firing very little. But the blue and the red are the cells that are cells firing in one sequence or the other, and there's absolutely no difference between them. So the cells, as far as the cells are concerned, they don't know the difference between a trajectory that goes to the left or the right until it happens. So the cells themselves appear to be stitched together to represent the environment. And the benefit of this is you have a basis for generalization. That means that if you've learned something about this trajectory and this trajectory, you might be able to make predictions about the never-before-experienced trajectory. So I want to finally get to the last part, which is one of Tolman's central ideas, was that sequences should be purposive. They should have reflect goals, and in the terms of these sequences, you should be able to simulate your actions to take you to a particular place. And I think it's fair to say this has actually been very difficult to show in hippocampal place cells. So just in place cell activity itself, there have been various reports that the cell's activity could be contingent on different contexts. But if you're correlating activity with a context, you have to repeat that context many, many, many times in order to get the data to say this cell prefers this context to that context. Whereas what we're looking for is a flexible system that can say, right, I want to get there. How do I get there? So our claim here is that the sequences themselves can be responsive. And that's been a very difficult thing to establish. I think it's fair to say that replay has generally been thought of as a sort of a spontaneous reverberation that might be spitting out all sorts of different sequences that you've experienced, but without any particular purpose, other than perhaps consolidating experiences you've had, but certainly not in terms of a directed navigational requirement. So we wanted to look at that specifically. We decided to look in a real spatial memory task, where we have a 2 meter by 2 meter arena with multiple possible goal locations, 36 possible goal locations. The idea was to get the animal to want to navigate to a specific location. And this way, this would give us the statistical power to be able to say if the sequences were approaching that one location as opposed to the others. The difficulty is, if the animal gets used to navigating to one particular location, he's not going to explore the whole environment. So we needed to come up with a task that would both allow for goal-directed navigation and exploration of the environment. So this is what we came up with. On odd trials, the animal has to forage around in this environment, and he doesn't know where the food will be. This chocolate, liquid chocolate, just pops up in one of these 36 little wells. Once he's found it, the very next trial, he's allowed to go back to the food. Then on the odd trial, the next trial, he has to forage around. Then he can go back to the food. So we're constantly toggling between random foraging trials and goal-directed trials. And the purpose of that is to get the animal to explore everywhere so we can get place fields everywhere. And we show here that the animal's much faster on the goal-directed trials than the random trials. So that establishes a number of things. That means he can't smell his way to the random trials. Otherwise, he'd be pretty fast at that too. And we know that he's motivated and able to remember where the food is because he's faster at getting there. The other problem we have to deal with is that we need a lot of cells. So you need something like between 10 and 20 cells arranged with fields along a linear track to be able to decode replay. So you're going to need something like 10 by 10 to 20 by 20, so between 100 and 400 cells to record in the open field. We actually don't have 400 cells, but we got pretty close. We did it by implanting 40 tetrodes simultaneously, bilaterally, so into both hippocampi. And so this is an example of the place cells we're able to record simultaneously. 212 cells, and these are their firing fields in this space. So you can see, firstly, let me draw your attention to these cells that fire everywhere. They're actually the interneurons, and they can be identified independently on the basis of the waveform and excluded. And the remainder of the cells have firing fields. These are all the cells that were responsive. There are other cells that we can identify in sleep, but they don't fire very much in the run, so they're not on the here. But the cells that are responsive have these place fields. So now that I've got all these fields, what am I going to do with them? I can't really show you these nice simple graphs anymore where I have cells on the y-axis and time on the x-axis because I've got a two-dimensional quantity. So I'm going to show you movies, so that's better. But how do I generate those movies? Well, I'm going to do the decoding. So I already showed you some data with this position decoding, but now I'm going to sort of explain the intuition a little bit more clearly. Essentially, what we do is we combine the spikes with the place fields. So we allow so every time, in a narrow period of activity, every time a cell spikes, it's kind of like voting for its place field. So imagine we have 4 cells that have place fields like this. If they all fire 1 or 2 spikes, I'm going to have a strong belief that this is the location where the animal is. What's important about this is that it doesn't have to be the current location of the animal. So if these cells are coactive, I'm going to have a strong prediction that the hippocampus is saying this is where the rat is, even though I know, because I have a camera, that that's where the rat is. But I don't always get a prediction either. So if the cells are firing in a non-coordinated way, I'm actually generating posterior probabilities of position, and I can say here the probabilities are very low for any given location. So now let me show you a movie or two. So what you're going to see here is the rat in the task. This is real time, and the rat, you can see his position and his head direction. It's exploring this maze. You can see the positions of the possible food wells. And here's the actual position that's baited.
Don't tip him off.
Yeah. And I'm going to have to play it again because I also want to explain what the flickering is. The flickering is the decoded representation in the hippocampus. There's a probability associated with every single pixel, but the fact that only a few pixels are brightly lit is because the representation is tightly focused. And in fact, this tight representation is much tighter than the size of any given place field. So the place fields are really cooperating during behavior to represent single positions. But what's perhaps more interesting is what happened next. And this is what happened just after this in the movie. But this movie is actually on a different timescale. I'll explain that in a minute. But this is what happened next. Then the position that's represented by the hippocampus smoothly translates away from his current position and moves off in the direction of the remembered goal position. Now, and that wasn't the same event repeated 4 times, that was 4 successive sharp waves, each of which contained this sequential activity. This was the remembered goal location. And time, if you had seen this, was moving much more slowly, because these are temporally compressed events, just like I said. So each of these events takes something like 100 milliseconds. Let me tell you how we analyzed it. So each frame of the movie corresponds to a short period of 10 milliseconds, during which activity is very tightly focused. And we haven't imposed that at all upon the system. That's a genuine feature, that at all moments during the events, the hippocampus seems to be very tightly focused in representing one position. Collapsing across time, we can now show you what the sequences look like. And here's just examples of the kinds of sequences we see just from one session. You see some of them are curvy, some of them are straight. But did they go anywhere? That's the real question. So I'm going to show you 2 things. Firstly, here's all the sequences that happen when the animal is at the home location. Now, there's something called an initiation bias. Awake replays tend to start in the current location. That could be because there's a very strong stimulus. It's obviously telling us something about the mechanism. I would say it's because it's navigationally useful to know how to get somewhere from here. It's not so useful to know how to get somewhere from somewhere else. But certainly, if you look across all of these events and just sum them all up, when he's at the home well, it's the home well that's in common. But what's interesting is if we now do the same trick for all the events that occurred whenever the animal was at a random well, the home well is also the event that's in common, suggesting that these events are converging on this remembered well. We can capture that, quantify that here, where we see that the total representation, meaning the amount of brightness in these probability figures, is concentrated at the remembered home well compared to all the other wells. And in fact, this is a statistical outlier. And the end point, if we just look at where the trajectories tend to end, that's also an outlier. So this is quite interesting because this means that as an experimenter, I can look at the activity during sharp waves when the animal's away from the goal, and I can tell you where the remembered goal is. And that's a kind of goal coding that we haven't been able to find before in place cells. And it's not because this location was more familiar. We can look at all the locations that the animal, for his own purposes, tends to enjoy to just sit in, and those locations that he doesn't spend very much time in, and they're just equally unrepresented compared to the goal. And then, what's important here is, I didn't tell you, but we actually changed the goal location that he has to remember every day. It takes him a few trials to figure out what that is, but we change it every day. Because if you remember, it's flexibility that we think is very important for cognitive mapping. And when we change the goal location to the new location, that becomes the overrepresented location the next day. So I showed you that the sequences start in the current location, end at the remembered location, but do they tell the rat anything about how to get there. So we did this analysis where we compared the future path and the past path, future path and past path with the path that the animal takes. Beg your pardon, let me start that again. The future path that the animal's about to take behaviorally, the past path that he's just taken behaviorally, and the replay sequence or sequence, place cell sequence that's occurred. And we ask simply, which of these two behaviors matches the sequence better? And we use this angular analysis to just say, what's the average displacement between the future path and the replay, or the past path and the replay? And we find that the future path tends to be very concentrated. These replays tend to be very concentrated at a short angular displacement with the replay. The replays tend to be very close in angle to the future path of the animal. They tend to be much less close to the past path of the animal. Interestingly, the prediction here is much stronger when the animal has to go to the home well than when the animal is moving somewhere else. So here, this is where the animal is at the home well, and he has to move off to do some random foraging. Of course, he knows where he's random foraging, but he knows where he's going to go. So he can predict what he's about to do. But the prediction is much weaker. And that could be for many reasons. It could be that there is a strong prediction, but then the animal changes his mind as he's randomly foraging. And then, interestingly, there seems to be this reverse, this sort of negative correlation here. And indeed, it was a feature of the task that the random wells never repeated. So perhaps the system knows that. So in the final thing I want to tell you is what I think is a very interesting aspect of this system, perhaps related to imagination. It's a feature of this task that we moved the remembered home well location every day, and we didn't repeat the random locations within the day, at least for the first 19 trials. What that means is that every single actual navigational act the animal was being asked to perform was a novel trajectory in terms of start and end location, even though it was a very familiar space. So I'm going to show you an example of, this is sort of a collection of different timescales in this movie, where you have behavior in real time, and then you switch to this replay time. But the point is to show you what the replay does when faced with this novel combination of start and end. So the animal's randomly foraging in this phase, and he's going to find the food, and then, hopefully, bang, you get a replay that starts where he is, moves nicely to the goal location. Then you get this interesting replay that goes down here. You can remember that. You get another one of these. Now this specific trajectory is not one that the animals ever had to make before. Then he does it for the first time. Then, interestingly, produces another sequence that also goes down here, just like that other one. And then he does. So that illustrates another thing that I won't have much time to tell you about, but it looks like there may be multi-step predictions in this activity. But this general trend for the sequences to match, to predict the future behavior, even when the specific sequences were novel, in this very familiar space, where they had to be sort of inferred, that was just as strong as any of the other replays. So, to sum up, hippocampal place cells represent the current location, but are also the substrate for simulating experience in other locations. The simulation sequences, I would say, exquisitely capture the spatial structure of an environment, and they are purposive. They're directed toward goal locations when an animal is planning his movements in a complex spatial memory task. So who was right all along? I'll thank Brad Pfeiffer, who's about to become faculty, starts as faculty at UT Southwestern in January for doing the spatial memory task, and Xiaoxing Wu, who worked on the Y-maze and my sponsors. Thank you.
I was trying to put together, I'm sure I misunderstand something, but there was a point early on in terms of solving the complexity of a multi-spaced and pathed maze, and you brought in the Markov advantage and similarly the sort of gold gradient feature. And so I would have thought in the end, with these unbelievable pictures of the projection, that it might have occurred in just the opposite direction. So what happened? What am I missing in terms of why it's not grabbing and coming to you, even though that's the next motion is for you to go there?
Right. I think because it's a little more complicated. So the phenomenology is that when the animal first experiences an environment, particularly we see this on linear tracks, you get a lot of reverse replay. And you get less and less, actually, as the animal becomes more experienced. The forwards replays, you start to see more as the animal becomes more experienced. I think that what's really going on in the open field task is he's able to use these forward replays are like hypotheses to be tested. And you often don't just get one, you see. You'll get, as you saw there in that example, you'll often get more than one, so it's as if the animal's testing two hypotheses and engaging other areas to say, well, is this a good thing I should do, or is this a good thing I should do? So I think that the forward sequences at this point in this task, which is very familiar with the task in other Go locations, represent, if you will, schemas or well-developed expectations. But early on in learning, I would expect there to be a lot of reverse replay. But the value function idea really works best when there's a fixed goal. So it could be just something about this task where the goal is actually moving and he has to be very flexible that means that strategy doesn't work so well.
Hi, thank you for your talk. I guess I have a question. I was considering sort of, say, this like linear maze where in some sense it's almost like a one-dimensional space where when he's walking down the hallway or whatever, there's only one direction he would ever consider going, and so there's really no value in making any predictions while he's walking at that time, versus like an open field where, you know, he could go in, it's like a two-dimensional space, and he could go in many different directions at a time. And so I guess I kind of have two questions. One is, do you see these ripples only when he's stopping? Because like you could think that the only time he might think in the linear maze is when he has to turn around or like decide to stay, versus the open field, and kind of what generally are the differences between what you see in an open field versus a linear track? So
let's see if I have this. So I didn't get the chance to talk about this, which is unfortunate because it's also very relevant. But you know, there's still controversies in the field very much, and so one of the things is what happens at a choice point. So you're right, linear tracks maybe not that interesting, but you can still build in choice points. So in that sense, you know, now the animal gets to a choice point, has to think what to do, and the theta sequences that I talked about could be a very good mechanism for just looking ahead if you've got some notion of value, or alternatively, you might, if you haven't learned it so well, you might need to have these longer extended sequences. So that's all we really know about the difference between running and stopping, is short sequences as long sequences. This was a result from David Regis's lab, which I decided, should I put this in, should I not put this in? He showed that when the animal gets to a choice point, the theta sequences will sweep first in one direction, and then in the other direction. This is tricky. These are decodings in successive short frames of where the hippocampal population, kind of like what I was showing, where the population says the animal is, and it sort of moves into one arm and then the other, as if the animal is considering what it should do next. So I don't see this as fundamentally that different logically from what we saw in the open field, except that here David is saying he sees these in theta. We don't get so much theta choice points. We think there might be more sharp waves, but that's a sort of ongoing sort of thing in the field. But what was very cool about it is that David Reddish associated this explicitly with vicarious trial and error, which is a term that Tolman coined to sort of indicate this sort of what looked like the rat thinking about what he might be about to do, sort of this kind of behavior. In fact, one of the examples was if the rat has to jump from a certain distance, before he jumps, he'll sort of make these little pretend jumps, as if he's mentally jumping to see if he can make it. So yeah, I mean, the Tolman's ideas are still influencing us. I'm not sure if I answered your question completely, but.
So have people looked at this in context remapping experiments to see if the sequences also remap, or are those preserved such that there's like a smooth warping of space?
That's actually a really, really good question. I mean, I would predict that. So there's no reason not to see sequences after the experience. And this is one of, like I said, it was first shown in sleep. And you can get sequences hours after the experience when the animal's in a completely different place when he's asleep. And even awake, you can get what are called remote replays of other experiences. So if there was a remapping, I don't see logically why you shouldn't get replays of the previous map. But where you're talking about partial remapping, where there might be some elements that are preserved and some generalization, anyway, I don't think we understand too much about that, and I don't know what the sequence will do, but I think it's a very good question.
So one of the things that you mentioned, Tolman said, was that overtraining an animal will turn it into like a stimulus response creature.
Yeah,
so
Lynn said that.
I was wondering, have you seen with overtraining that these sweeps towards the goal go away and the animal just walks over?
Yeah, it's a really good question. It's actually experimentally quite tricky because if the animal's highly motivated and gets into a stereotype behavior, it doesn't stop. And so experimentally, we can't say whether he would or would not, the hippocampus could or could not generate these sequences. So it's hard to disentangle the behavioral changes. Like, for example, on the linear track, once the animal becomes highly trained, he just eats and goes. And so, you can overtrain an animal to do hundreds of trials on the linear track, but then you won't see replay sequences. But I don't think the capacity to generate them has necessarily disappeared. But it's difficult.
One question about the replay events and sharp waves in general, and sort of how you think replay at the time scale, a very short time scale of a few hundred milliseconds, can actually, be involved in a cognitive operation like planning and thinking about where you're going to go. Sort of, do you think other events are happening at a slower time scale in different brain regions, or how does that work?
That's good. So I think I mapped out something in the reverse replay, where the events would set up some kind of plasticity where you associate place cells with value, and then when you actually are in those place cells fields, you get that access to that value. So that makes sense. What you're asking, I think, though, is what about the forward sequences? How can that carry over into something like a movement plan? And I don't know. I mean, obviously, that would imply there was some kind of short-term memory. It's also possible that it doesn't initiate a plan, but is also some kind of offline learning, and the animal's just sort of testing how well he understands the environment in some sense. The experiments that need to be done are being done by at least, I think, 2 labs, and they are to try and online detect the sequences and interfere with them, and interfere with sequences that approach that reflect one trajectory but not another. That's a very, very challenging thing to do, but actually not impossible. And you don't have time to go into it, but there are some tricks. But basically, you can actually make good guesses online about what the replay is, the information content of the replay. And so that's the hope, that we can do those experiments to show a causal role. And then we'll worry about how it does it.
Okay, I think we better move on to the last talk now. People want to stand up and give what they want.